Assalamu alaikum. My name is Hajra Yunus and today's lecture we will discuss about mutation. In the today's lecture we will cover the topics: what is mutation, endogenous sources of mutations which are replication error, slippage error. In replication error we will discuss about slippage error and base substitution, and then topoisomerase mediated mutation, transposons mutated mutation, tautomeric shift, spontaneous depurination and deamination. So first is, what is mutation? As mutation is the change or alteration in the genetic material or DNA of an organism. As you can see in this diagram here, this part of the DNA is normal, whereas this part of DNA has mutation. Okay. So mutation, as you can see here, is the change or alteration. It's the normal part of the DNA and it's the altered or mutated part of the DNA. Okay, if there is a change or mutation or alteration in the genetic material or DNA of an organism and which is permanent, it is called mutation. As you know that if there is a damage in DNA, if there is a DNA damage, so in case of DNA damage in our body or in the body of organism, repair machinery is present, and repair machinery is responsible to remove the damage. If the damage is removed, and then you can say it's the temporary damage. Okay, but if damage is not removed and it's permanent, so it will lead to mutation. Okay, so if the DNA damage is permanent, permanent means if the DNA damage is not removed by repair machinery and the DNA damage becomes part of next cycle of replication, so in that case it is permanent, or if it is permanent it will lead to mutation. These changes can occur spontaneously or be induced by various environmental factors. We will discuss later which environmental factors are responsible for or are responsible to induce mutation. Mutation can have a significant impact on the characteristics and evolution of living organisms. As you know that the evolution is because of mutation. Okay, it is not necessary every time mutation is negative. Okay, mutation may lead to diseases if mutation is negative, and mutation may also lead to evolution if the mutation is positive. Okay, so mutation can have a significant impact on the characteristics and evolution of living organisms. A mutation was discovered by Hugo de Varis in evening primrose plants. Next is the causes of mutation. There are two different causes of mutation. One is the endogenous mutation and other one is the exogenous mutation. In case of endogenous mutation, internal factors within an organism that lead to DNA change. So internal factors are responsible for endogenous mutation. As you can see here, error during DNA replication, spontaneous chemical agents such as deamination or depurination we will discuss later, and various other reactive oxygen species, and there are many more. And exogenous mutation, external factors are responsible for change in DNA. For example, radiations such as UV or X-ray radiations, and there are two other radiations like beta rays, gamma rays, are also various chemical agents such as alkylating agents and intercalating agents, and various biological agents are also responsible for exogenous mutation. Biological agents such as certain viruses, because viruses come from the outside and they are responsible for causing mutation from outside. Now, first we will discuss about endogenous causes. So in case of endogenous causes, first is the error in DNA replication. Okay, if there is a process of DNA replication and error in DNA replication, so it can be base substitution and slippage error. Okay, And in case of base substitution, it can be of two types. Number one is the transition, another one is the transversion. First we will discuss about base substitution. Okay, so what is base substitution? As you can see here, a base substitution mutation is a type of genetic mutation where one base pair in the DNA sequence is replaced by another. Okay, if only one base pair in the DNA is replaced by another, so it is called the base substitution. As you know that the base substitution is of two types. Number one is the transition, another one is the transversion. So now, what is the difference between transition and transversion? As you can see in this diagram, in case of transition, uh, pyrimidine is converted into pyrimidine and purine is converted into purine. Okay, so you know that the pyrimidines are thiamine and cytosine, and purines are adenine and guanine. So in case of transition, one pyrimidine is converted into another pyrimidine, or one purine is converted into another purine. As you can see here, T is converted into C and A will convert into G. But in case of transversion, pyrimidine is converted into purine and purine to pyrimidine. As you can see here, thiamine and cytosine, these are the pyrimidines. Thiamine will convert into, let me change the marker, adenine and guanine, and similarly cytosine will convert into guanine and adenine. And here the purine, you know that there are two purines, adenine and guanine. Adenine will convert into thiamine and cytosine, and guanine will convert into cytosine and thiamine. So these are the two different types of base substitution. In case of base substitution, only one base is converted into another base. And there are two different types. Number one is the transition, another one is the transversion. As you can see here, in case of transition, purines to purines and pyrimidines to pyrimidines. And in case of transversions, pyrimidines to purines and purines to pyrimidines. Now there is one question, that out of both, out of transition and transversion, which one is the more deleterious, or which one has the deleterious effect? So the answer is transversion has the deleterious effect. Deleterious effect. Okay, so why transversion has the deleterious effect? Because in transversion, pyrimidine is converted to purine and the purine to pyrimidine. Okay, so you can say from single ring structure to double ring structure and double ring structure to single ring structure, whereas in case of transition, single ring structure is converted into single ring structure and the double ring structure is converted into double ring structure. So out of transition and transversion has deleterious effect or is fatal. Now we will discuss about DNA replication errors. As you can see in this diagram, this is it's the strand of DNA without any error. As you can see here, complementary to adenine, thiamine is present. Okay, now in this strand, you know that during the process of DNA replication, the two strands get separated. So it's the first template and it's the another template strand. Okay, so now here complementary to thiamine, adenine is present. Okay, so it's the copy of the parental strand, so there is no problem, it's the correct strand. But you can see here in front of adenine, now here guanine is present. Okay, if the damage is repaired by the repair machinery, okay, if the damage is repaired by the repair machinery, so there will be no problem. But if the damage is not repaired, you can see here in the second round of replication, and you know that these two strands get separated. This is the one parental strand and this is the another parental strand. So you can see here in front of adenine or complementary to adenine, thymine is present, and here the complementary to guanine, cytosine is present. But you can see that in the actual parental strand, adenine and thymine were present, and here adenine and thymine were present. But you can see in this, after second round of application, in this strand, instead of adenine and thymine, guanine and cytosine are present. So you can see this is the error during the DNA replication. So if the error is repaired, so there is no problem. But if the error is not repaired, so it will become part of second round application, then third, fifth, fourth, fifth, and so on. Okay, so it will become part of next generation. Next is the another type of base substitution is the nonsense mutation. What is nonsense mutation? The change creates a stop codon. This is very important, stop codon, leading to the premature termination of protein synthesis. As you can see here, it's the original strand of DNA, and you can see in original strand of DNA all amino acids are present. You can see here the amino acids are present, and as you can see in this diagram here, the open reading frame of the protein is the large in size. So this is the open reading frame. So what do you mean by open reading frame? Open reading frame is the frame which is visible to you, okay, that starts from the start codon and ends before the stop codon. Like you can say that the open reading frame of the protein starts from the methionine and ends just before the stop codon. Now you can see here, so after 1 2 3 4 here, instead of cytosine or a single amino acid, okay, so in case of the base substitution, only one amino acid is replaced or is changed. As you can see here, only C is replaced by T, and because of the replacement, firstly the amino acid was present, and now because of the replacement of just one amino acid, instead of C now T is present, so TAG codes stop codon. Okay, this is for stop codon, and this TAG is the stop codon that does not code anything, does not code any protein. It shows that now the protein of synthesis of the protein has stopped. And now you can see there is one more thing, change in the size of open reading frame. As you can see that in the original DNA strand, what is the size of open reading frame? That was large in size, but because of the change in size of the open reading frame and because of the presence of stop codon, you can see now the open reading frame has reduced. Okay, and because of the reduction in size of open reading frame, there is a truncated protein. What is the truncated protein? Truncated protein is the premature and protein with the small size or with the short length. Okay, and if there is a truncated protein, that can be non-functional. So you can see that the nonsense mutation will lead to the will affect the open reading frame, lead to the production of truncated protein, and in that case the protein can be non-functional. So in that case, nonsense mutation has deleterious effect. Okay, next is silent or same sense mutation. In case of silent or same sense mutation, the change does not affect the protein because the new codon codes for the same amino acid. Okay, so this is very important thing when it comes to the silent or same sense mutation. So as you can see, this is the original strand of DNA, okay, and it's the after mutation. Okay, I would not say it's the mutated strand, but it's after mutation. And here different amino acids are present, like tyrosine, tryptophan, leucine, valine, histidine are present. In case of, and you know that the silent or same sense mutation is another type of base substitution, and in base substitution only one amino acid is changed. So here TAT, in case of TAT, T is replaced by T. Now TAC is present, and you know that for tyrosine is coded by TAT and TAC. C. Okay, because you know that in one amino acid is coded by more than one codes. Okay, similarly in case of tyrosine is coded by TAT and TAC. So if for before tyrosine was present and after mutation the tyrosine was present, so there is no change in the amino acid sequence of the protein, so you can say that it's the same sense or silent mutation. So the first tyrosine was present, after tyrosine was present, and there is no change. So it is also said that the silent or same sense mutation is conserved in nature because it will not affect the the structure of protein. Okay, because in case of silent or same sense mutation, the what amino acids are present before mutation, the similar amino acids are present after mutation. The only difference is that the change in codon. As you can see here for tyrosine, TAT was present, and after mutation instead of TAT, TAC is present, but it will not affect the functioning of protein, it will not affect the open reading frame of the protein, it will not affect the structure of protein. So everything is silent or same sense. Next is missense mutation. In case of missense mutation, the change results in different amino acids being incorporated into the protein, which can affect its function. So it is called missense mutation. As you can see here, in case of missense mutation, here the methionine, glutamic acid, alanine, glycine are present, and after the missense mutation, in case of missense mutation, only one amino acid. Okay, this is also type of base substitution. So here GAA was present, and instead of A, now here C is present. So GAA codes for glutamic acid, and now GAC codes for aspartic acid. Just because of change in one amino acid, the whole amino, or change in just one nucleotide, the whole amino acid gets changed. So you can see here the protein structure was normal, but here you can see the abnormality or mutation in the DNA strand. Let's take the example of sickle cell anemia. In case of sickle cell anemia at 6th codon, so you know that in case of sickle cell anemia, hemoglobin is affected, and in case of hemoglobin, beta chain is affected. Okay, and in case of beta chain at position number 6 codon, so normally GAG is present. Okay, and this GAG is for glutamic acid. If glutamic acid is present, it's the normal protein, but after mutation, in case of sickle cell, adenine is replaced by uracil. Okay, if adenine is replaced by uracil, now instead of glutamic acid, valine is present, and now it is called the mutant protein. Why? Because in case of sickle cell anemia, if there is mutation at codon number 6 and instead of glutamic acid valine is present, it will affect the functioning of hemoglobin. And you know that hemoglobin is responsible for carrying the oxygen to blood or different parts of body. So if valine is present, so ultimately it will result in the affecting the functioning of hemoglobin. So this is the missense mutation. So in case of missense mutation, silent mutation and nonsense mutation, only same sense or the silent mutation does not have deleterious effect because there is no change in structure of protein. But in case of missense mutation and nonsense mutation, they both have the deleterious effect because of the change in structure of protein and change in the size of open reading frame. Next, we will talk about insertion or deletion. Okay, it can be another type of base substitution. In case of insertion or deletion, if there is a single or double nucleotide addition or deletion, it will lead to frame shift mutation. Okay, now we will discuss about base deletion or base insertion. Okay, firstly we will discuss about base deletion. As you can see, it's the original strand of DNA, and that has leucine, arginine, histidine, serine, aspartic acid, arginine, various other amino acids are present. But if at this point, if this A adenine is deleted, so you will see that the whole operating frame has been shifted or has been changed. So here the leucine was present, still leucine is present because the deletion was after that. So here the deletion was, and after deletion on, you can see the whole operating frame has been shifted. Now here the arginine was present, here the now glycine is present, here histidine, here another amino acid, and you can see the operating frame after deletion has been shifted. So deletion has the deleterious effect on the DNA structure. Next is the base insertion. As you can see in base insertion, in case of original strand, after leucine, the one amino acid, you can see here, the one amino acid has added. So because of the addition of one amino acid, you can see again the open reading frame has been shifted. Here the leucine was present, and before the insertion arginine was present, now glutamic acid is present, and histidine was present, now alanine is present, and you can see the whole operating frame after that has been changed. So if there is an insertion of one nucleotide or double nucleotide, it will lead to frame shift mutation, and from where the nucleotide is deleted, or where the nucleotide is inserted, and after that it will lead to frame shift mutation. Next is the another cause of the endogenous mutation is the slippage error. Okay, in case of slippage error, generally occurs in the telomeric region. Okay, so but you know, exception is everywhere, so generally it is in the telomeric region. So as you can see in this diagram, why it is in the, if it is in the telomeric region, so you can see in telomeric region the repetitive sequences are present, you know that. Okay, and now here the DNA polymerase is present, which is responsible for the replication of telomeric region also. Somehow DNA polymerase slips and falls, and when it is, and you can see here the DNA polymerase is present. Okay, and sometime it happens that the DNA polymerase falls down, and when DNA polymerase is reloaded back, so at that time, because of the repetitive sequences of the telomeric region, so DNA polymerase may get confused, so where to attach, because the all sequences are similar, they are repetitive sequences. So you can say here, instead of loading here, so if the DNA polymerase gets loaded here, so you can see here this extra sequence is present, this extra piece of nucleotide or this extra piece of DNA has been polymerized, so it will lead to loop formation. Okay, so there is a loop formation. Why there is a loop formation? Because when the DNA polymerase falls down or is loaded back, at that time the DNA polymerase may get confused where to bind, because telomeric region has repetitive sequences, and in that case the TLA polymerase gets bind on the back, so you can see here the extra piece of DNA is present, so this extra piece of DNA will convert into the loop. So now there is a loop. But in case of the telomeric region, if the loop is in telomeric region, so there is no problem. Okay, it will not affect the functioning of the protein, it will not affect any other thing. But you know that exception is everywhere. If this extra nucleotide or this loop region is in the genomic region, so what would happen? It will lead to the unstable DNA structure and various other symptoms also. As you can see, let's take the example. So CAG repeats. If CAG repeats in the nuclear laminar region, they are present. Okay, and if CAG repeats are more than 40 in number, so it will lead to one disease which is called the Huntington disease. Okay, in case of Huntington disease, the symptoms are the, or you can say the Huntington disease will lead to unstable DNA structure, chromatin at their not at their fixed place, or increasing aging process. Okay, so you can see that if the slippage error, or means you can say the DNA polymerase falls down at the telomeric region, so there is no problem if there is a loop formation. But if the DNA polymerase falls down in the genomic region, and here the CAG repeats are more than 40 in number, you can see if there is a loop formation, or if the CAG repeats are in nuclear laminar region and they are more than 40 in number, so it will lead to the various other disease. And we studied about or talked about one disease, that's the Huntington disease. Okay, next is the topoisomerase mediated DNA damage. You know that the topoisomerase is responsible to remove supercoiling from DNA strand during DNA replication. You know that DNA strand is highly supercoiled structure. Okay, to facilitate the process of DNA polymerase, DNA helicase, topoisomerase is responsible to induce cut and then ligate the DNA strand and remove the firstly induced cut, remove the supercoiling, and ligate the DNA strand. So topoisomerase is responsible for these things. But for example, if there is a mutation in topoisomerase and topoisomerase just cut the DNA strand but not ligate, okay, so topoisomerase just cut the DNA strand but there is no ligation because of the mutation in topoisomerase, so it will also affect the process of DNA replication. Okay, so topoisomerase mediated damage is in the topoisomerase. Next is transposons mediated damage. As you know that the transposons are the jumping gene, they can move from one place to another place. And for example, if the transposons, as you know that it's the sequence of DNA, and here intron and exons are present. Okay, so is the intronic region and exonic regions are present. And for example, here it's the transposon. So it is the transposon gene, and if this gene jumps from one intronic region to another intronic region, so there is no problem, because as you know that during the RNA splicing, introns are removed, only exons are present. So if the transposon gene is jumps from one intronic region to another intronic region, so there is no problem. But if the transposon jumps from one exonic region to another exonic region, so in that case it will affect the structure or function of the gene, so and it will lead to the insertional inactivation. As long as transposons are in the intronic region, they will not cause any error. But if the transposons are the exonic region, it will affect the structure of gene and it will lead to the addition, and you know that addition or insertion is deleterious or has deleterious effect, and it will lead to the insertional activation. Next is tautomeric shift. In case of tautomeric shift, as you can see that, what is tautomeric shift? Tautomeric shifting is the shifting of hydrogen ions into nitrogenous base. Okay, as you can see here, it's the cytosine and it's the keto-guanine and it's the thiamine and enol-guanine. As you know that guanine forms bond with cytosine. Okay, and guanine forms, and between guanine and cytosine there are 3 bonds. As you can see in this form that the guanine is present in keto form and the cytosine is present in amino form. And in case of, and here another thing, as you can see here, the cytosine has the amino group, and here the one extra hydrogen is present and the guanine does not have. So here it acts as a donor and it acts as a acceptor. Okay, now here you can see, so these dots are for the acceptor and the straight line is for the donor. Okay, so dotted line is for acceptor and the straight line is for donor. So here it's the donor and acceptor and donor and acceptor. Now you can see tautomeric shift is the shifting of hydrogen ion into the nitrogenous base. Okay, now you can see there is a shifting of hydrogen ion. Okay, if the hydrogen ion gets shifted, so you can see here the keto guanine is converted into enol guanine. Okay, so now the keto form or keto guanine is converted into enol guanine, and keto guanine only forms bond with amino or cytosine. Guanine only forms bond with cytosine, but here the not the keto but enol form is present. If there is enol form, so enol form cannot form bond with cytosine. Now why? Because you can see that here the guanine has acceptor, donor, but you can see here the donor, acceptor and donor, and you can see that here the position of bond has changed, and because of position of bond, change in the position of bond, you can see now this enol form of guanine cannot form bond with cytosine. Now it will form with thymidine, because here acceptor, donor or acceptor, or you can see the enol form of guanine will form bond with that which has acceptor, donor and acceptor, and thymine has all these things. So in that case enol guanine will make bond with thymine. So in tautomeric shift, the first thing is shifting of hydrogen ions into the nitrogenous base, and second important thing is that the keto form is converted into enol form. Okay, this is very important thing. So whenever it's a question about tautomeric shifting, so tautomeric shifting is the shifting of hydrogen ions into nitrogenous base. So as you can see here, so there is, after shifting of hydrogen ion, the keto guanine is converted into enol guanine, and now enol guanine cannot form bond with cytosine, it will form bond with thymidine. And you know, and there is one more thing, in case of tautomeric shifting, it leads to transition. How it is transition? As you know that the purines are adenine and guanine, and pyrimidines are cytosine and thymidine. Okay, so as you can see here, firstly with the keto guanine, cytosine was present. Okay, and after the tautomeric shifting, there is a enol form, and enol form forms bond with thymidine. So you can say the firstly cytosine was present, and after that thymidine was present. So this is the transition. Okay, we studied about that, the what is transition. In case of transition, one form pyrimidine is converted into pyrimidine, purine is converted into purine. As you can see here, the pyrimidine is converted into purine. The first cytosine was present and the cytosine was replaced by thymidine, so it's the transition. Next is spontaneous depurination. In case of spontaneous depurination, the purines leave the DNA strand. As you know that by some estimate, As many as 10,000 purines bases are lost by depurination each day in a mammalian cell. And in some books it is more than 18,000. Okay, so you know that the purines are the double-stranded DNA structure, and the purines are very heavy, and they leave the DNA strand, or they leave the DNA strand more than 18,000 times per day, and they come back. For example, so it's the DNA strand, and here the purine was present. Okay, so you can see here adenine was present, and here thiamine is present. So because adenine is the purine, so now at the time of DNA replication these two strands get separated, and here thiamine is present. So in complement to DNA strand, here adenine will form, or adenine DNA polymerase will attach adenine. But here there is another strand of DNA, and for example here different amino acids are present, but here purine is not present. As you can see here, the adenine is present, but at the time of DNA polymerization, if adenine is not present, so DNA polymerase will not add anything. So you will see that there is a deletion of one strand, because DNA polymerase will not attach anything, so it will lead to the deletion. So deletion, in case of deletion, there is the uh, the open reading frame, size of the open reading frame is affected, and you know that deletion has the deleterious effect. So depurination, if there is a depurination and the amino acid is not at the usual place, and at the time of DNA polymerization, DNA polymerase does not find anything, DNA polymerase will not attach anything, it will lead to the deletion. Okay, let's take this example. As you can see here, it's the original strand of DNA, and here there is a depurination of amine of adenine. Okay, there is a depurination of adenine, and now you can see here the AP site is present, means here the purine is not present. Now at the time of DNA replication, you can see the size of the, or the length of the DNA strand. The one strand has the is long, and the one strand is short. So one strand is normal, and one strand is the deleted strand, or one strand has the mutation. So this is the effect of depurination. Next is the spontaneous deamination. Okay, in case of spontaneous deamination, there is a deamination, removal of amino group from the cytosine, adenine and guanine. Okay, cytosine, adenine and guanine have the amino group. If the amino group is removed, so you will see now cytosine will convert into uracil, adenine is converted into hypoxanthine, and guanine is converted into xanthine. And now because of the change in structure, the affinity of bonding will also change. First, you know that cytosine forms bond with guanine, adenine forms bond with thiamine, and guanine will form bond with, or guanine forms bond with cytosine. But because of the change in affinity, now here uracil is present. Uracil will form bond with adenine. Hypoxanthine will form bond with cytosine, and xanthine will form bond with cytosine. As you can see here, because of the removal of amino group, now the structures have changed, and the change in structures, you can see the affinity has also changed. For now the cytosine is converted into uracil, and the other partner will be adenine, and here the hypoxanthine will form bond with cytosine, and xanthine will form bond with cytosine. So if we summarize, in today's lecture we discussed about what is mutation and what are the endogenous sources of mutation. So in endogenous sources of mutation we discussed error in DNA replication, and which can be due to the slippage error, or base substitution. We also discussed about spontaneous depurination, spontaneous deamination. And in base substitution, we discussed about different types of base substitution that can be transition or transversion, and there are other types of mutation which come under the category of base substitution, that are missense mutation, same sense mutation, and nonsense mutation. And we also discussed about tautomeric shift. I hope you understand the lecture. If you have any questions, you can ask me in the comment section.